answer is, it's time for another lesson. Hey Bio-20s, I'm back again for another lesson, and today we are going to finish up talking about aerobic cellular respiration. So just a recap of what we learned last video, we learned that aerobic respiration requires oxygen to go through. There are 4 stages involved in this process. So we have glycolysis, pyruvate oxidation, Krebs cycle, and then oxidative phosphorylation, or the electron transport chain. Glycolysis is a very complicated 10-step process that occurs in the cytoplasm. The oxidation of pyruvate, Krebs cycle, and oxidative phosphorylation, all 3 of these processes require oxygen, and all 3 of these processes take place in the mitochondria. So that's very important to note. These 3 are happening in the mitochondria, glycolysis happening in the cytoplasm. So just a recap of our last video, we talked about glycolysis, where we take 1 molecule of glucose, we are going to add 2 molecules of ATP to produce 4 molecules of ATP for a net gain of 2 ATP. We're also going to make 2 NADHs, which we'll save for later, and we're going to have 2 pyruvate molecules at the end of this, each having 3 carbons. So we have 1, 2, 3, 4, 5, 6 carbons to begin with. Now we have 2 3-carbon molecules. So what we're going to end up doing is taking each pyruvate, running it through a round of what we call pyruvate oxidation, which will occur in the mitochondria, and then taking each of those products and running it through a round of Krebs cycle. So let's look at pyruvate oxidation first. What we are going to do is we're going to take each pyruvate molecule, we are going to remove one of the carbons to make a CO2. So we're going to do this for each pyruvate. So ultimately we'll be making 2 CO2 molecules. And to make the CO2, we are going to need some available oxygen. So if oxygen is available, we can move to the next step, which is pyruvate oxidation, which will occur inside of the mitochondrial matrix. This will be inside the mitochondria. Okay, we are also going to make some more NADH, so we're going to make some more electron carriers, and we are going to finally take the remaining 2 carbon molecules, attach a coenzyme onto it, and create a compound called acetyl-CoA. So it's basically a 4-step process here. So what we're going to do is we're going to take our pyruvate molecule, which is a 1, 2, 3 carbon molecule. We are going to remove one of those carbons. So I'm going to take one of those carbons off and I'm going to remove it. I'm going to stick it with 2 oxygens. There's one of my CO2 molecules. Now remember, because we have 2 pyruvates, everything we are doing here is going to be multiplied by 2. So we're making 2 molecules of CO2, and we're making 2 molecules of NADH. And once again, we're just going to take that NADH, that electron carrier, we're going to set it to the side for now. We're not going to use it just yet. We're going to set it to the side for step number 4. And then for each pyruvate, we're going to stick on what's called a coenzyme A to produce a 2-carbon acetyl-CoA. So at the end of pyruvate oxidation, you've made 2 acetyl-CoAs, 2 CO2s, and 2 NADHs. How many ATP? 0. We didn't actually make any ATP in this process. So if we go back to the first step, glycolysis made 2, pyruvate oxidation made 0. Overall, we're trying to get to that 36. Majority of the ATP is going to be made during the last couple of steps. Okay, each acetyl-CoA is now going to enter the Krebs cycle. This is also taking place inside of the mitochondria. What we're going to do is we're going to run the acetyl-CoA through a series of reactions similar to Calvin's cycle, where there was that series of steps happening along the way. Same idea here. We're going to run the acetyl-CoA through a cycle of reactions that will produce 3 NADHs, 1 FADH2, 1 ATP, and then 2 CO2s. Notice how this happens twice because there are 2 acetyl-CoAs. So let's take a look. So we're going to run our pyruvate through the Krebs cycle. When we do, we actually remove the coenzyme A and it gets recycled to be used again in the upper step of pyruvate oxidation. We're going to take the final 2 carbons and attach them to oxygen to make CO2. Now, since this is happening twice, once again, you got to remember this is happening twice because there's 2 acetyl-CoAs. you end up getting a total of 4 CO2s. So just think about it. We've made 4 CO2s during Krebs cycle, 2 CO2s during pyruvate oxidation, for a total of 6 CO2s. Hence why there's 6 in the balanced equation of cellular respiration. Okay, we're gonna make 2 NADHs here, 1 NADH up here, for a grand total of 6 NADHs. And we're also gonna make 2 FADH2s, which is similar to NADH. They're both electron carriers. It just has a little bit of a lower energy than the NADH does. And then finally, we're gonna make 1 molecule of ATP, but once again, since we're doing it twice, it ends up being 2 molecules of ATP. And now we are all out of carbon. We've used up all the carbon, but we've only made 4 ATP, which doesn't seem very great, does it? We've made a lot of electron carriers, yes, and now it's time to put those carriers to use. Because at the end of Krebs cycle, we are out of carbon, so all the carbon has been released as CO2. The coenzyme A is gone, it's going to be helped to reuse, and now the hydrogen that we've been making, as well as the electron carriers, are going to allow us to complete the final step, which is basically making a bunch of ATP. So the final step is called oxidative phosphorylation, or electron transport chemiosmosis, which you might remember those terms from photosynthesis. Okay, so in this last stage, we are going to make the majority of ATP using those electron carriers that I've told you to put to the side, the NADH and the FADH2s. We are finally going to put them to use. So what they've been doing is they've been transferred up to the end here of the inner mitochondrial membrane, inside that mitochondrial matrix. So here's the inside of the mitochondrial matrix. Here's the inner mitochondrial membrane, and here's the intermembrane space. Now you're going to notice something that's going to look really familiar. We are going to take our electron carriers, like our NADH, and we were going to donate electrons to an electron transport chain, very similar to photosynthesis. And we're going to move those electrons down a transport chain, 2 electrons for every NADH, until they reach an oxygen molecule. Now remember, we haven't really talked much about water. Where is water coming from in all of this? Well, actually, this is where the water is produced. The oxygen is going to gain electrons, it's going to be reduced, as well as grab a couple hydrogen to make H2O. This is where we're making the water. Now, how do we go about making the ATP? That's the biggest question. Well, when the electrons move down the electron transport chain, hydrogen is actually going to be pumped out of the matrix because the electron is going to pull the hydrogen with it, and the hydrogen is going to be attracted to that negative electron. And what we're doing is building a gradient outside of the mitochondrial membrane matrix. Now we're going to have a bunch of hydrogen outside that want to get back inside, and the only way they can is through an ATP synthase complex, which will create ATP, essentially slap a phosphate onto an ADP, creating ATP. This entire process is essentially harnessing those electron carriers we created in step 1, 2, and 3, taking the excess hydrogen ions, pumping them out of the mitochondrial matrix, and creating a gradient, electrochemical gradient. And then once again, nature hates gradients, always wants to even them out. The only way to even them out is to then pump the hydrogen into the matrix again. By doing so, we make ATP. So once again, sound familiar? Very similar to photosynthesis. Okay, so how much ATP do we actually get? Because once again, we get 2 from glycolysis, 0 from pyruvate oxidation, and 2 from Krebs cycle. This is where the remaining ATP comes from. Each NADH has enough energy, enough electrons to produce roughly 3 ATP molecules. It's actually a little bit less, but for simplicity purpose, we're just going to say 3. So for every NADH you make, you can essentially say that every NADH is worth 3 ATP. And then FADH2 is a little less energy. It actually starts a little bit further down the electron transport chain, so it's only going to net us 2 ATP. And finally, those electrons, this step is called oxidative phosphorylation because the electrons, right, are then being added to the oxygen. So being added to the oxygen to then make water. Oxygen needs to be readily available to accept those electrons. If it's not, you cannot do this last step. So to keep the electrons flowing, oxygen needs to be there at the end to accept them, grab a couple hydrogens, make water. In total, you're gonna make 6 molecules of H2O. Okay, so then I like to end with this balance sheet because it kind of ties everything together very nicely. So it kind of shows you where all the 36 ATP are coming from. So we make 2 just in glycolysis, so there's 2 ATP. We end up making 2 NADHs, but in order to get the NADHs from the cytoplasm into the mitochondria, they actually have to be converted into FADH2s, which will reduce the number a little bit. Now, some textbooks won't include this part. They'll say it's 38 ATP versus 36. Both numbers are acceptable because at the end of the day, it's more of a theoretical yield. In actuality, you're looking at probably like, I don't know, 28, 30, maybe 32 ATP, but nowhere near 36 or 38. That's like in a perfect world. So when we see an FADH2, we're just gonna think 2 ATP, right? So these 2 are worth 2 ATP each for a grand total of 4 ATP. In pyruvate oxidation, we made 2 NADHs, which are worth 3 ATP each for a grand total of 6 ATP. In Krebs cycle, we made 2 ATP, we made 6 NADHs worth 3 apiece, so that's 18 ATP, and then we also made 2 FADH2s worth 2 apiece. If you add up all the ATP that you created, you get 36 molecules of ATP. And that's where our glucose goes. If oxygen is present, you'll run through glycolysis, pyruvate oxidation, Krebs cycle, ETC chemiosmosis to get 36 molecules of ATP, which can then go into your cells to do all those cool things like move your muscles, pump things in and out, and all that other stuff. So next lesson, we're going to talk about what happens if oxygen isn't present and how will this process differ. So thanks again for watching, and we'll chat soon.